Hi everyone. So this is our last chemistry lecture, and this lecture will end module 1. So after you complete all of the chemistry and intro materials, you'll be ready to take the first exam. In this lecture, we're going to talk about lipids and proteins, the other two large biological molecules. We already did carbohydrates in chemistry part 4. And then the fourth group is nucleic acids, which we're going to do a little bit later in the semester. So first we'll talk about lipids, which are also known as fats. Some people distinguish between them as lipids being mostly in animals and fats in plant foods, but that's not universally used that way, but they are interchangeable, both the term lipids and fats for the way we're going to use them. Probably fat is a term you're more familiar with. The other thing that's a little bit confusing about lipids is that as a group, they're not as well organized as like carbohydrates, where we had monosaccharides, disaccharides, and polysaccharides. What they have in common as a group, lipids, is that they're hydrophobic. Hydro is water, and phobic means fear. They don't actually have a fear of water, but they won't dissolve in water, so they're insoluble in water. And so that's their common characteristic. So they're not organized more as much for their structure like carbohydrates is, as for the fact that they're hydrophobic. For animals, fats are our primary way we store our calories. So when we consume more calories than what we need, we convert the extra calories primarily to fat to store. So it doesn't matter, we get calories from carbs, protein, and fat are the three sources of calories in our diet. So if we eat extra carb or extra protein, we don't really store them that way. We convert them to fat and store them as fat. That is actually a more efficient way of storing calories, because for every gram of fat you store, you get 9 calories, but for every gram of carb and protein you store, you only get 4 calories. So it's more efficient to store them as fat, because again, they take more than twice as much energy as carbohydrates or proteins. They're organic molecules, mainly carbon and hydrogen with a little bit of oxygen, but not as much oxygen as what we saw in carbohydrates. And they're non-polar covalent bonds. So they don't have a charge like water does. Remember, water was polar covalent. So that's why they're hydrophobic. They don't dissolve in water. If you put oil and water together, if you shake it real hard, they'll mix up a little bit, but then as soon as you let them sit again, they'll separate. One of the primary functions then of fat or lipids is energy storage in animals. And then it's also insulation to keep our body temperature, help regulate our body temperature. So under our skin, we have a layer of fat to help regulate body temperature, and also to protect most of our internal organs like the heart and the kidneys. They all kind of have a fat cushion around them to help insulate and protect them. When you talk about types of lipids, we're not going to talk about all of these in details. Fats and oils, again, it's not always differentiated that way. They're actually similar structurally, but fat, they're referring to animal products, and oils to plants. So things like corn oil, safflower oil, olive oil would all be plant oils, whereas fat, like butter, lard, the fat around a steak, would all be animal fats. We're going to talk about both of those. Phospholipids we're going to talk about in a week or so, because they're important variations of fats that are found in our cell membranes. Steroids, I'll talk about a little bit, and waxes, things like earwax and other types of wax, which are mostly in plants. I'm not going to really talk about them. So fats, when we use the word fats and oils, those are the same type of molecules. They're triglycerides. So you can get from the word, the tri part of that, it means there's three of something. And what a triglyceride is, it's a molecule of glycerol, which is shown right here, with three molecules of fatty acids. And that's what forms a triglyceride. And this is the way all of our fat is stored. So if you were to take a sample of fat from one of your fat stores and analyze that, it's almost all triglycerides. So we store our extra calories as predominantly triglycerides. And so we make a triglyceride with fatty acids and glycerol through dehydration synthesis. So we remove the water and then the fatty acid gets attached to the glycerol. Glycerol is actually made from glucose. If we need to use some of our stored fat, we would break it back down through a hydrolysis reaction where we add the water back and remove the fatty acid from the glycerol. So dehydration synthesis and hydrolysis are used to make and break down triglycerides. I wanted to talk a little bit more about the fatty acids. So fatty acids are the long chains that are attached to the glycerol. And there are two basic types of fatty acids, unsaturated fatty acids and saturated fatty acids. And I'm going to, instead of reading the words here, I'm going to go to describe it. So fatty acids are a long carbohydrate chain. So this one has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 carbons. So this one is a 17 carbons long. They range from like 8 carbons to 20 some carbons. So one of the things that vary from one fatty acid to another is the length, the number of carbons that make up the fatty acid. The other thing is the absence and presence of double bonds. So if you look at this fatty acid here, between the carbons, there are no double bonds. They're all single bonds. Remember, single bond you indicate with just a single dash. So fatty acids that have all single bonds are called saturated fatty acids. And saturated fatty acids are found primarily in animal fats. So things like butter, lard, again, the fat in meat are more saturated fatty acids. And because of that long straight chain, they tend to pack more solidly. So they tend to be more solid at room temperature, not a hard solid, but things like butter. They're not really oils at room temperature. The other type of fatty acids are unsaturated fatty acids. And unsaturated fatty acids have one or more double bonds. So you can see here is a double bond, here is a double bond, here is a double bond. So this fatty acid has 3 unsaturated double bonds. So unsaturated fatty acids have one or more double bonds. Sometimes if they have more than one, they call them polyunsaturated. If it's just one, they're called monounsaturated. And they're found mostly in plant foods, and they're mostly oils. So things like safflower oil, corn oil, olive oil tend to be higher in unsaturated fatty acids. And the reason they're more like oils is that these double bonds make like a kink in the molecule. And so because of all those kinks, they're not able to pack together as solidly. So they're looser, and that's why they're more like a liquid. And you may be aware that saturated fatty acids, a diet high in saturated fatty acids, which again are more animal fats, is associated with an increased risk of coronary heart disease, whereas diets high in unsaturated fatty acids have less risk of coronary heart disease. So there's been a move to encourage individuals to decrease their intake of animal fats and replace it with increased use of plant fats. So again, a triglyceride is glycerol with 3 fatty acids, and the fatty acids can either be saturated or unsaturated. The 3 don't have to be the same fatty acid, so you can have an unsaturated and a saturated. And again, that's the way we store our fat, and it's also the primary fat that we eat because we're eating animal or plant stores of fat, so we primarily eat triglycerides. So if we eat a triglyceride, it goes through hydrolysis to remove the fatty acids, and we absorb them as individual fatty acids from our small intestine. If we have extra calories and we convert them to fatty acids, we make triglycerides and store them in our fat. And as I mentioned, that the saturated fatty acids are associated with increased risk of coronary heart disease. And this just gives you relative risk. So like the normal risk would be 1. So anything that is above 1 would be a greater risk, and anything that's below would be a less risk. So you can see that a diet higher in saturated fat increases the risk of heart disease. And these two are, this is monounsaturated with just 1 double bond. These are poly with more than 1. That lowers your risk of heart disease. You may have heard of trans fatty acids. They've been mostly removed from foods in the past 10 years or so. And trans fatty acids are kind of mostly an artificial type of fatty acids that were produced in processing of foods. For example, like making margarine. So margarine is made from plant oils, which makes it a liquid, but it wants to replace butter, which is a saturated animal fat. So in order to get it more solid like butter, it went through a processing that converted some of the unsaturated fatty acids to a different form called a trans fatty acid. And that made it more solid-like and therefore more like butter. The problem with that is that they've discovered that trans fatty acids actually have a greater effect on increasing your risk of heart disease than the actual saturated fatty acid. So in recent years, there's been a move to eliminate trans fats from processed foods. And so lots of times you'll see on labels that a food contains no trans fatty acids. I remember going into Whole Foods one day and they had a sign in the front of the store that the whole store had no trans fatty acids. So they've been slowly being eliminated from foods sold in this country. So those are the triglycerides, which are the fats and oils. Another type of lipid is steroids. And steroids, I'm not going to really talk about waxes, so eliminate that one. But steroids, there are a number of steroids that are important physiologically. Steroids are cholesterol and related compounds. So anything that kind of has that stero as part of the name is a steroid. And so cholesterol is one steroid that you're probably familiar with. A diet high in cholesterol is also associated with an increased risk of coronary heart disease. And routinely, if you have blood work, they'll measure your blood cholesterol level. And if it's high, they'll recommend ways of lowering your blood cholesterol level. Lipitor, for example, which is one of the primary drugs used to lower blood cholesterol level, I think is the most prescribed drug in the United States. So cholesterol we kind of think of as being a bad guy, but it actually has physiological functions. The thing is, we don't really need to eat it, so you can eliminate cholesterol from your diet and not have any problems because you can actually make cholesterol in your liver. And so cholesterol has this kind of complicated chemical structure, and one of the primary functions of cholesterol is that we use it to make steroid hormones. And steroid hormones are things like testosterone in males, estrogen and progesterone in females, cortisone. Those are all steroid hormones that are made from cholesterol. And so testosterone is the primary male sexual hormone that turns an embryo into a male, like a male peacock here. And estrogen is the primary female one, which that's a female peacock. So steroids have important physiological functions, and again, they're all made from cholesterol. Okay, so now we're going to switch from doing lipids to proteins, and that's the final of the biological molecules that we're going to talk about now. So proteins are the most complex of the three that we've done, of the carbohydrates, lipids, and proteins. And they are large polymers of various sizes, and they're made from monomers called amino acids. And so amino acids are the building blocks of proteins. And we have 20 different, well, all living things have 20 different amino acids. The amino acids all have certain characteristics that are the same, and then some that make them slightly different so that they have different properties. So let me structure of an amino acid. So this is a basic structure. So all amino acids have a central carbon atom, and attached to that carbon is a group called our carboxyl group, which is a carbon, two oxygens, and a hydrogen. And then the other group is an amino group. And an amino group has nitrogen with two hydrogens. And this is the first time we've seen nitrogen. Carbohydrates and fats do not have nitrogen. So if I show you a structure and say, is this a carbohydrate, fat, or protein, and you see nitrogens, then you know it's going to be an amino acid and not a carbohydrate or fat, because they do not have nitrogen. So that all, and then there's a hydrogen here. Remember, carbon can have 4 bonds. And so all amino acids, all 20 of them, have the central carbon, the amino group, the hydrogen, and the carboxyl group. And then the fourth group is what is different. R doesn't stand for a letter. It means the variable group. I don't know why R means variable, but it does. So the 20 different amino acids have different things attached at the R site. So I'll show you the 20 different amino acids. So these are all 20 of them. And so if you look at any one, they have the central carbon, the carboxyl group, the amino group, the hydrogen, and then for this one, this is the simplest amino acid. The fourth, the R group, is just an H. If you look down here at this one, here's the central carbon, the carboxyl group, the hydrogen, the amino group, and then the R group is this very complicated structure attached here. So the 20 different amino acids, and highlighted in the light blue is what the R is, have different R groups, and that gives them different chemical characteristics and functions. You may have heard of some of these. Tryptophan is one that a lot of people have heard of because it's used to make the neurotransmitter serotonin, which is thought to be related to feeling full and sleepy. And so that's tryptophan. And we'll be talking, you don't have to memorize this list or anything like that, but we'll be talking about these in more detail when we get to some of the functions of proteins and how proteins are made. Because proteins, we have lots of different proteins in our body. We have more than 75,000 different proteins. And the reason we can have so many is that these 20 amino acids can be combined together by dehydration synthesis to make lots of different proteins. It's kind of like the letters of the alphabet. We have 26 letters of the alphabet, and we can put them together to make millions of different words. Likewise, we can put these 20 amino acids together, different order, different lengths, to make lots of different proteins. So we're going to be talking a lot about different proteins and the importance of proteins and what your genes carry. And we'll be talking more about this as we go through the semester. What your genes are your instructions for making all of your 75,000 proteins. So that's what you pass on to your offspring, is it's like a cookbook for making proteins that is inherited from parents. So when you're making a protein from amino acids, you join two amino acids together from dehydration synthesis to form what's called a peptide bond. So that's the bond between two amino acids. And the way that works, it's described there, but I'm going to picture. So here's one amino acid, and here's another amino acid. So we take the OH from the carboxyl group, and the H from the amino group. We bring them together. That's what forms the water. And then this carboxyl group bonds to the nitrogen in the amino group, and that's what's called a peptide bond. And then another amino acid can come along and attach here, and another one and another one. So you can have, some proteins have more than 100 amino acids, some have fewer. Insulin, for example, is a protein. It has, I think, 31 amino acids. Hemoglobin, which carries oxygen in your red blood cells, has 130 amino acids. So the length of proteins varies greatly. Some of the amino acids, you need all 20 of those amino acids in order to be able to make all of your proteins. So you make, as I said, you have 75,000 different proteins inside of your cells. So you need to be able to make them. And so the reason we need to eat protein, when we eat a protein through hydrolysis, it's broken down to amino acids. Those amino acids get into the blood, into our cells, and then we use those amino acids to make the proteins that our cell makes. So we need to have all those amino acids available to us inside of our cells so we can make the proteins that our cells need. And so for the amino acids, some of those we have to eat, like we can't make them. So we have to eat them, and those are called essential amino acids. But some of the amino acids we can make from other amino acids. So if we don't eat them directly, we can convert other amino acids. So 8 of the 20 are essential that we have to have in our diet. And so we have to eat proteins that provide those 8 essential amino acids, because we don't have the ability to make them. And if we don't have them, we can't make our body proteins, so that would cause malfunctions in our body and protein deficiency. So you have to have those 8 animal fats. The best protein to meet the requirements for our essential amino acid is actually egg. Chicken eggs are considered the perfect protein because they have the amino acids that we need, the 8 essential. Generally speaking, animal proteins better provide our essential amino acids because kind of all animals have the same essential amino acids. So therefore, by eating animal proteins, we know we're getting our essential amino acids. However, you can get all the essential amino acids by eating combining other proteins that might be missing one or other of the amino acids if you're eating only plant proteins. And some of the kind of common foods that people eat actually are called complementary proteins because they combine all the amino acids. So rice, for example, is missing, I can't remember if it's 2 or 1 of the essential, and beans is missing 1, but it's not the same one that rice. So when you put them together, you get all of your essential amino acids. So that peanuts and bread, this is yogurt, but yogurt is milk, so it's made from milk, so it has all the essentials. So you can get all your essential amino acids by combining. So if you're not eating animal foods, and if you are just eating plant foods, you have to be more aware of combinations of proteins to be able to get all your essential amino acids, particularly in young children who are growing and have a higher protein requirement. Proteins form these long chains of amino acids, which is what is showing here. And then those chains fold in specific ways. So even though this picture here looks like you took a piece of rope and tangled it all up, it's actually a very precise folding of those amino acid chains to give you the structure of the protein. And that folding is essential for that protein to be able to work properly. This protein here is hemoglobin, which is the protein in red blood cells that carries oxygen. So it's very essential for you to be able to survive because it takes oxygen from your lungs to every cell in your body where it's needed. And so there are conditions where hemoglobin doesn't work properly, and because it doesn't fold properly, and you have problems in getting oxygen delivered to your tissues. So the folding of the protein is essential for it to work for whatever function that particular protein has. If you destroy the folding of the protein, it can no longer do what it's supposed to do. That breaking down the protein shape is called denaturation. Sometimes we do that, things like cooking, temperature can do that. Like when you cook an egg, for example, the protein albumin is in the white of the egg, and before it's cooked, it's kind of like a transparent fluidy, and then you cook it and it gets white and solid. And that's primarily due to denaturation of the protein, changing its characteristics, but it would also change its function if it happened in a cell. I'm going to give you an idea of how diverse and important proteins are in humans. I'm going to talk about, but it's not just humans, it's in all living organisms. I'm going to just briefly go over some of the types of functions that proteins have in our bodies, and to give you an idea of both their diversity and importance. So these are the 6 types of functions. I'm going to just briefly mention each of those. Go to the next slide. So structural proteins, those are proteins that are important in giving shape or structure. For example, collagen is an important protein in skin. Tendons and ligaments are made primarily of collagen. So they're like strong kind of rope-like proteins that are important in giving shape and support in various parts of our body. Transport proteins are proteins that carry things from one part of our body to another. So we mentioned hemoglobin, which is in red blood cells, and it carries oxygen throughout your body. You have other proteins that carry lipids. So we have a number of transport proteins that are used to carry specific nutrients from one part of the body to the other. Movement, the way our muscles are able to contract are through the sliding of proteins over top of one another. And if you take anatomy and physiology, you'll learn about that in detail. So the ability to move is mostly due to functions of specific types of proteins. Our defense or our immune system, antibodies, which are made by white blood cells, not red blood cells that transport oxygen, are proteins. And so in order to be able to make antibodies, you need to be able to make proteins. So that's another type of protein that's important biologically. Many hormones are proteins. We talked about steroid proteins like estrogen and testosterone, but other proteins are, other hormones are proteins, like insulin is a protein, FSH is a protein that's involved in stimulating the testes and ovaries. And then a final category are enzymes, and enzymes control chemical reactions inside of our cells. And breakdown of foods are all controlled by enzymes. And we're going to have a whole lecture on enzymes in a week or two. So we'll talk much more about the functions of enzymes when we get to that point. So that's the end of our biological molecules presentation. And that's the end of the material covered in Module 1. Bye-bye.